Hi, so my name is Hassan Amir. I'm going to be just in general explaining my study titled the Clinical Grade Genomic Analysis of a 99 patient cohort for the Identification of Therapeutic Targets. So essentially the project revolves around personalized medicine, genomics, and everything in that kind of realm. And so what essentially the study aims to do is be able to provide clinical insights into advanced metastatic disease settings for specific cancer patients. And I'm from Calgary, Alberta, here in Canada, and I'm excited to present my project.
Do you have a presentation you're going to share?
Yep.
Rock and roll. And Hassan, we see that.
Okay, sounds good. So my project is titled the Clinical Grade Genomic Analysis of a 99 patient cohort for the Identification of Therapeutic Targets. I've done this in correspondence with Dr. Pinaki Bose with the Department of Oncology at the Arnie Charbonneau Cancer Institute at the Cummings School of Medicine here at the University of Calgary. Problem: tumor genesis and conventional treatment. Conventional treatment has shown limited success within clinical practice as they do not take into account the individual molecular profiles of cancer patients. For example, we have three major types of conventional treatment that's usually used in treating cancer patients, including chemotherapy, radiotherapy, and surgery. Now, chemotherapy essentially involves cytotoxic agents that disrupt the biological functions of cancer and has generally leads to several negative implications such as toxic potency, low tolerance levels, and acquired cell resistance. You have radiotherapy, in which you essentially damage DNA through the use of ionizing radiation, which tends to lead to cell arrest and cell death, and can also harm normal healthy cells and generally leads to acquired cell resistance as well. Then you have surgical applications. So this is heavily dependent upon clinical data, such as the stage of cancer, the patient demography, and tends to be in general highly dysfunctional in advanced metastatic cases. And what it generally does is it essentially reduces it reduces the quality of life of the cancer patient and leads to multiple negative complications such as bleeding, infection, and scarring. Research: next generation sequencing and comprehensive molecular profiling. So advanced metastatic solid tumors are usually characterized by tumor heterogeneity, meaning you have distinct mutations all in that same tumor type. Now, not all of these mutations actually contribute to cancer. 99% of all mutations are passenger mutations, meaning they do not contribute further to cancer. However, 1% of those mutations are driver mutations, meaning they're contributed for and further contribute to cancer. If we can identify those specific driver mutations, we can slow down cancer, we can slow down the progression of cancer and increase prognosis rates within cancer patients. So if we can identify this, we can essentially lead to higher prognosis rates within cancer patients, and that's the fundamental rationale of my project. Then you have prognostic outcomes. Once again, if we can identify specific driver genes or specific biomarkers, this allows for the prediction of disease progression through the identification of specific biomarkers. And if we can predict disease progression, then we can implement specific therapeutics and specific drugs in targeting that cancer within that specific cancer patient. Then you have treatment resistance. I talked about chemotherapy and radiotherapy. These different therapeutics tend to be highly resistant as treatment is added. So what essentially happens is that our cancers in our body can become resistant to chemotherapy and radiotherapy. Now, this is also associated, this kind of resistant mechanism is also associated with specific mutations, specific driver genes, and molecular pathways. So we can identify these. We can target these specific molecular pathways, these specific mutations, and these specific driver genes, and using specific targeted therapeutics, which will essentially enable the development of effective treatment. And so kind of the use of NGS, which is essentially a DNA sequencing technology, it allows for the development of therapeutics personalized/tailored to the individual cancer patient. You have to realize chemotherapy and radiotherapy are kind of blanket treatments. We can apply them to all cancer patients, but if we can take a cancer patient's individual molecular profile, analyze it, and then based upon that analysis, use specific drugs to target those specific discrepancies within his cancer, that will lead to positive prognosis outcomes within cancer patients. That leads to my research question. Using Illumina's TST-170 panel to sequence a set of 99 tumor samples, can cancer driver mutations be identified, provide insight into targeted therapies and personalized medicine? Rationale, methods, and objectives. Essentially, the rationale was to identify driver genes in a cohort of cancer patients to provide insight into potential therapeutic targets. In terms of my methods, I developed a bioinformatics pipeline to conduct comprehensive DNA mutation analysis in a cohort of 99 cancer patients. So this involved exploratory data analysis, ClinVar analysis, cosmic analysis, and map tools analysis. This was all done through Microsoft Excel and R programming language applications. Furthermore, I also quantified RNA expression within the Genotype Tissue Expression Project. So this is a specific database that contains information on normal RNA expression of specific genes when they're during that fundamental process of transcription. You have the Cancer Genome Atlas, which is another landmark genomics project, a $3 billion project that has essentially molecularly characterized over 133,000 tumors and really is able to provide that kind of general standard benchmark of our data analysis. Then you have our R data set that we specifically implemented, the TST170, and this was done to essentially use the GTEx as a threshold to identify overexpressed genes, which may further provide secondary analysis in identifying specific driver genes. So this is my objectives. Firstly, I wanted to profile DNA mutations, specifically stratify our data set based upon frequency, variant type, and amino acid changes. Furthermore, I also wanted to conduct ClinVar and cosmic analysis. So ClinVar is essentially a database that provides that essentially provides analysis on the clinical significance of specific variants. Then you have Cosmic, which is another database that provides information on the frequency and distribution of variants across the human genome. And this acted as our secondary analysis. Then we implemented a specific bioconductor package known as map tools within the R programming language. And that is essentially implementing that map tools package really allowed for that holistic analysis and really allowed for more advanced and complex analysis within our general pipeline. Then finally, we conducted RNA expression analysis using those three main databases, the Cancer Genome Atlas, Genotype Tissue Expression Project, and our dataset denoted as TST170 throughout the presentation. So data collection, Illumina's TST170 panel, and secondary analysis. So a cohort of 99 cancer patients underwent tumor biopsy resections at the Tom Baker Cancer Center, located here in Calgary, Alberta, here in Canada. The tumor biopsy samples were stored as formalin-fixed paraffin-embedded tissue specimens, specifically the FFPE samples, and there was 99 of them in total. Then we implemented a specific next-generation sequencing assay or a specific method called the TrueSight 170 panel. The TrueSight 170 panel is essentially a DNA sequencing platform that analyzes 170 genes known to be pathogenic in both RNA and DNA, as reported by previous literature. And so this was essentially performed on the FFPE samples, which outputted FASTQ file types. Now, these FASTQ file types provide insight upon DNA mutation analysis. And we specifically used Illumina's NextSeq 550 sequencer, which was a really comprehensive sequencing platform provided by Illumina, which is a common biotechnology provider. These FASTQ file types were then used as inputs in Illumina's Dragon semantic pipeline, and they outputted VCF file types. So VCF file types provide information on RNA expression analysis. Then we conducted secondary analysis using two major tools common within the industry. We used Sophia DMEM Genomics, and essentially we annotated and pre-classified variants from our raw TST170 NGS data outputs. We also used another software known as Integrative Genomic Analyses, in which we essentially visually explored our NGS data and determined whether a mutation was true or whether a mutation was false. So we can kind of move on to our first kind of general results here. So within our general exploratory data analysis, we will see the following 5 genes as the top frequently mutated genes within our data set. So for example, we had BARD1, TP53, ATM, Notch1, and CHEK2. And so after determining these as the key genes of interest, we essentially conducted our ClinVar and cosmic analysis. So as I said before, ClinVar essentially looks at the clinical significance of these specific mutations within our general genes. So I can name a few results here in our ClinVar analysis. Within TP53, we will find 55% of all genes to be pathogenic, which indicates that this does further contribute to cancer, further does contribute to the tumor growth and dispersal. Therefore, this is a good gene in general to target. Our cosmic tissue prevalence analysis, we looked at the frequency and distribution of these variants across the human genome. We were able to find around 14 variants within the large intestine tissue. And so within our final conclusions, this gene was deemed pathogenic and was to be further analyzed later in our pipeline. Another example would be BARD1. BARD1 is perhaps not the best gene to further target using therapeutics. As you can see, around 40% of it is benign, meaning it does not further contribute to cancer and wouldn't necessarily be the best gene to further investigate and to further target. Within our cosmic tissue prevalence analysis, we were able to see around 2 variants present within prostate tissue, and it was deemed likely benign due to its due to the fact being that almost half of all of its genes were benign. And then we can move on to kind of like some interesting genes. For example, Notch1. Within Notch1, we were able to see around 50% of all variants to be of uncertain significance, meaning if we can investigate this gene further, we can perhaps find specific novel findings that we can indicate back to these public archives of the ClinMar Cosmic Database. So you can see around 50% of all variants there were deemed of uncertain significance, and around 2 variants were found within both the UAT and large intestine. So this is our kind of general results number 1, and general kind of looking at the clinical significance of our exploratory data analysis results. We can move on to results number 2. So we essentially determine the somatic mutational landscape within our data set, specifically within lung adenocarcinoma, breast cancer, and ovarian cancer. So what you see right here is kind of the first plot you see is the oncoplot. The oncoplot generally provides a holistic overview of our data set, providing coding sequences, providing specific genes of interest, and also providing sample ranges and the TMP metric as well. So as you can see, we analyze the top mutated cancer variations here, breast, lung, and ovarian. We were able to find specific genes of interest, specifically being AR, ATM, BARD1, CHEK2, and ALK. And we were able to find around 76% of all samples related to breast cancer, lung cancer, and ovarian cancer. So that indicates that perhaps these are the best cancer variations to look at within our data set, as they're the most commonly frequently mutated and common variations. And we were also able to see our sample range. So sample range was between 1 to 34. So as you can see here, AR within our oncoplot has a sample range of going from 1 to 34, indicating that this gene is present within 34 different samples or 34 different cancer patients. That's a really high number. If we can develop specific treatment strategies for that specific gene, then this may lead to high prognosis rates within multiple cancer patients, in this case being 34. Then the plot you kind of see to your kind of the blue plot there is our exclusive / co-occurrence event analysis using our TST-170 data analysis. So we essentially performed a specific pairwise Fisher's exact test, which essentially looks at the co-occurrence of 2 data sets in a contingent, which looks at the co-occurrence of 2 variables in a contingent data set. And so this was done to look at gene pair detection. And we use p-values of 0.05 and 0.1, which essentially indicate how significant our results really are. And so what this provides insight into is another class of drugs known as double target agents. Double target agents are specific drugs that target one or more molecular pathway or driver gene. So if we can essentially target, so this is another, so usually within cancer, when we have 2 driver genes within a single tumor, this leads to angiogenetic effects within that cancer, within the gene. So that further enhances its ability to circumvent multiple processes such as apoptosis and immunosurveillance processes as well to further contribute to cancer. So as you can see here, more than 95% of genes co-occur within our data set among all major cancer variations, meaning that we can really implement double target agents and specifically double target class therapeutic drugs to further increase prognosis rates of cancer patients. Specifically, we will see MSH3, CHEK2, BRCA1, BRCA2, and PIK3CA to exhibit the greatest number of co-occurrence within our analysis. Then we can look at the general mutated regions of interest and specifically hotspots. So as you can see, we looked at the frequency and distribution of different genes across our protein transcripts. So we will see, so this essentially allows us to determine the functionality of genes within our data set. So for example, we have AR here. As you can see, the androgen receptor is significantly mutated. And so since the androgen receptor is significantly mutated, that would mean that this is a loss of function gene and that this further contributes to cancer. And this was specifically found within lung cancer. Another one, if you look, you can see BRCA1. There we will see a mutated protein domain, specifically in the BRCT domain, but and that was deemed a loss of function mutation, meaning it does not further contribute, meaning its final protein product has a loss of function, which further contributes to cancer. Otherwise, here in EAN3, but you can also see within BRCA1, you can see the EAN3 protein domain, and that's significantly mutated. And previous scientific literature essentially reports that EAN3 is associated with plant growth. And so to see that in human patients within our cancer variations indicates that this is perhaps a novel finding, a novel protein domain that further contributes to tumor growth and dispersal. Finally, our final results here are of our TP53, Notch1, and kind of general other pathogenic genes, and we conducted RNA expression analysis. So the Genotype Tissue Expression Project here in orange, as you see, is essentially a database that provides insight upon normal RNA expression. So when you have a mutated gene and you have a normal gene, that when that mutated gene is transformed into RNA through that process of transcription, it tends to have lower RNA expression. But when it's mutated, it tends to have higher RNA expression. So if we can identify higher RNA expression using the GTEx as a threshold, we can determine upregulated and downregulated genes. And if you have upregulated genes that further contribute to cancer, as reported by a seminal research paper called The Hallmarks of Cancer. So as you can see here, you're able to see multiple genes here. For example, TP53 common in the literature, but we were able to find one novel finding specifically within our FLT3 gene. FLT3 RNA count overexpression was significantly higher within our breast cancer variation, and that was reported only twice in previous literature.
Hassan, I'm sorry to interrupt you. This is fascinating, and you've done a lot of work. We have about 1 minute left before the next presenter, if you can kind of wrap it up.
Yeah, okay. So we essentially looked at multiple drug candidates here. We determined multiple genomic drivers, and we essentially looked at multiple oncogenic mechanisms. So we looked at multiple drug candidates here, which all have shown success within clinical practice. So you have afatinib, you have dasatinib, and you have vemurafenib. These are all small molecule inhibitors that further inhibit molecular pathways associated with tumor genesis. So kind of conclusions and applications here. So I believe that the study has potential for a publication, and all R programming applications are available on GitHub, a public hosting services for further research purposes. A couple limitations would be a small patient cohort and just the general access to our clinical data. If we add access to sex, age, gender, race, and ethnicity, this may have all acted as significant statistical indicators and may have provided further insight for clinicians and patients as well. So in terms of conclusions, the study serves as an excellent reference on the effectiveness of Illumina's TSC-170 panel. It provides excellent insight into prognosis and potential positive clinical outcomes, and it further understands the underlying causes and mechanisms of tumor genesis and provides novel evidence of certain drivers. I'd like to express my gratitude and thanks to mentor Dr. Pinaki Bose, product supervisor Reid McNeil, and everyone else at the Bose Lab. Thank you for listening.
Thank you, Hassan. That was a lot of data.
Yep.
Enormous amount of data in a very short amount of time.
Yep.
But you did a lot of really outstanding work there.
Thank you.
So I don't know if we have any questions from YouTube, but I don't see any. Although I feel like I would have to be a doctoral PhD to have one at this point.
Okay.
Amazing. Nice job.
Thank you.